So we continue from the previous class to summarize by the tyrosine biochemistry. The first one is the mechanical properties providing by the examples of resilin. Second one, GFP, green fluorescence protein. And then the third one is light absorbing protein, that is photoactive protein. This is final the examples of tyrosine, which is melanin. So the melanin is your pigment, actually. So depending on the amount of the melanin you have, and then it becomes more dark and dark brownish color. And no melanin, as you know, that white skin you have it. So the melanin is actually generating from the melanocyte. So as name indicated, the melanin generating cell. The myocyte, right, is like a muscle properties presenting cell, like fibroblast fiber presenting cell, generating cell. Like this is melanocyte. So cyte means cell. So the melanin is basically, it's complicated, but you have start from tyrosine, and this is a C alpha carbon, and then there's H and CO minus, I would say, and H3 plus. So you start from here. There is an enzyme called tyrosine hydroxylase. Tyrosine hydroxylase, and that change like this. So this is the dihydroxyl. So name is 3 and 4 dihydroxy. So if you remove this, then what is it? Phenylalanine, right? Dihydroxy phenylalanine. So we call this very famous amino acid DOPA. We probably don't know the DOPA. DOPA is a drug for treating or delaying the progress of the Alzheimer or the Parkinson's disease, actually. So very old people who has the Parkinson's disease frequently injected by this DOPA. I think everyone knows it's dopamine. Dopamine, that's brain and neurotransmitter. So dopamine is out of this carboxyl group. This carboxyl group out by dopa D carboxylase. So the carboxylate group out. This is enzyme that. So now the dopamine structure. So then 1 carbon, 2 carbon, and then you got an NHCH2 because they decarboxylated, and then NH2. So this is the famous dopamine. So you actually start from the tyrosine. So then you can imagine that the melanocyte heavily generating tyrosine, amino acid. Okay, then amino acid becomes dopa and dopamine, and now what happens? See what happens. So the absorption, this now, remember the first cases that called the dityrosine, which is the resilient structures. We learn about dityrosine. That is quite a closely related, that chemistry and this chemistry. So I will continue this one. So there's electrons are out. Typically 1 or 2, but typically I would say the 2 electrons out. So actually out from this amino acid. Also same as, say, 2 electrons out. I'll do this one first. When right, this is the electrons are out, right? So this is a quinone structure. This dopamine. Quinone. So quinone is what is CO double bonds. That is how we call them quinone. So this is dopamine quinone. So it's similar to dopaquinone. So dopaquinone is right, and then got this. So this is dopaquinone. What happens is these are chemically reactive because they lose electrons. They lose electrons means that these would like to obtain electrons back. So they want to have electrons back to have this very stable pi structures. This benzene structure. Benzene is the most stable structures in organic chemistry, right? So if they want to have electrons, so now see what happens in how they get electrons. Actually, if you have this one, see if I throw this like this NH2, then there is electrons here, right? So that's why they have either reacting intramolecularly intramolecular reactions. Same as here, same as dopamine quinone. So this is dopaquinone. Dopaquinone and dopamine quinone share the same chemistry. The same. So here we have, like, I redraw this. Then we have like this. So then they form. What should I? Okay, I'll draw like this way. Draw like. So this one reacting to here. So then we got. Like this, right? So this is a cyclization reactions. There's a further oxidation, so it becomes increasing double bonds, right? So then we got this is a further oxidation losing electrons again. Then we have this is dihydroxy in both. If I don't want to draw this dopaquinone cases because all you have to do now is adding the carboxyl group here. So again, carboxyl group here. And then that now carboxyl group here, carboxyl group here. So this is the case of the dopa case, right? So now this becomes a melanin precursor. This is a melanin precursor now. So I will redraw how the melanins. So now see what happens is all the melanocyte does is actually providing a heavily amount of the tyrosine, and there's a tyrosine keeps secreting and making the tyrosine, and then there's an enzyme located, and then they becomes quinone, dopa or dopamine, and then just let them go for the oxidation of this process, and then finally it becomes indole structure they form. All the spontaneous reactions, okay, the spontaneous oxidations. And then if I start from here, and then we start from, say, so this is the 5, 6. So this is we call the indole 5, 6 and 5 nitrogen. We call indole structure. At the point of 5 and 6, dihydroxyl. So it's 5, 6 dihydroxy indole DHI. So now, same here. Electrons oxidated, and actually the oxygen in the cell becomes got electrons. This generating the reactive oxygen species actually. So electrons are taken by the oxygen nearby. But you don't need to think about this part. But anyway, so if you have questions about where the electrons are located, so this is the answer. So then now. So then now we have 1, 2, 3, 4, 5, 6. Okay, so then now we have quinone again. Okay, so we have some strange in here. Okay, so we have now. If I draw this, sorry, redraw this. Okay. So this is the dopamine, the DHI, quinone. So this is the quinone, and then they lose electrons. So what happens is this quinone is chemically reactive, as I mentioned before, chemically reactive, unstable, would like to have electrons back, right? So then they now have to cross into each other. So if you have another quinone is nearby, same molecules is nearby because the cells are keep producing tyrosine. So now they have another DHI quinone comes reaction, so they becomes a dimer. The dimer is that we have now. Six now, same. Somehow it's strange. 1, 2, 3, 4, 5, 6, and then we got something like this. This dimer of the DHI is formed because they take the electrons from this nearby molecule. So now we change a little bit topic. These chemistries occurs in dityrosine. So this is dityrosine. Same. So tyrosine we have now C alpha, H, H2, here, right? And then lose the electrons, becomes, well, I'm not drawing all the sequences, but got a quinone here, right? And then there's another molecules nearby. The quinones is formed. electrons is back. Same as here. Same as the chemistries that via quinone structures by the oxidation process. So this is the resilient chemistry now. So delete this part. So now the topic is not here. Why? So melanin basically we know that the color is brownish and dark brownish color, right? So let me begin with so we know that in 20 different amino acids we have absorption of the UV absorption at 280 nanometers is by tyrosine, tryptophan, and phenylalanine. Right? Phenylalanine is this, right? And then this tyrosine is anyway 280 nanometers, and then this becomes, if I throw like this way, then the electrons are conjugated, so they can actually travel through this molecule. So they lose the energy. That becomes about 320 nanometers. Now you got a dimers out of it. Then electrons are traveling through all this molecule, losing more energy. So that becomes approaching to 400 nanometer absorption. Now you can imagine that this another DHI quinones are reacting to the existing DHI dimer, and becomes a trimer, trimer becomes tetramers like this. And then these, probably these two dimer species absorbing this 400 nanometer species. What if you can imagine here, if this guy, say the trimers maybe 470 nanometer. I'm not sure the exact values of the light absorption, the wavelengths. This one maybe 5 or 520 nanometers, 600 nanometers, something like this, right? See what happens now. So what happens? This is absorption. This is say 300 nanometer, 400 nanometer, 500, 600, 700, something. This is our wavelengths, which is nanometer. And then when a molecules that absorb all the visible light becomes black, right? Black color, because they absorb all the light. White means all the reflected, right? The light. So see, in this one about 300 absorption, 70 absorption, then you sum everything's molecules, all the mixtures of dimer, trimer, tetramer, pentamers, and wall mixtures. Then you got all absorptions until this. Visible wavelength reach. So that's why it becomes dark. Okay. So this is from what? From actual tyrosine structures. Okay. This is why simply providing the tyrosine from the cell by the melanocyte, and then they form very dark pigment. So this dark pigment is very good for protecting from the light. So then you will say whatever UV, also they protect the UV region all the light comes to the skin, and then the melanin actually absorbs the light, and no effect at all through this the epi and endo, a little bit thelials, the region, and epi and endodermal region located this pigment, and then below it there is critical cells, but there's no damage because they were all absorbed on the light damage from this environment. Okay, this is the melanin. So melanin is from the tyrosine. So this is a fourth case, GFP, resilin, light absorbing protein, which is PYP, and the melanin. This is all the tyrosine biochemistry part of it. Okay, so this is the end of the tyrosine biochemistry class, and then I hope that you enjoy my class. Also, I will just upload it, the random, the educational, the YouTube, and also related to biochemistry, also related to others. I hope that you can, from time to time, you can come back to my channels and then see increasing your knowledge of the biochemistry. Okay, thank you very much. Bye bye.